Modern scientists say that nuclear medicine is the key to quick cancer treatment. Over the past 3 years, Russian researchers have developed more than a dozen innovative drugs that can safely and quickly cure malignant tumors. The International Agency for Research on Cancer estimates that cancer incidence will increase by 50% by 2040. It sounds scary, but experts attribute this to better diagnosis. Cancer takes the life of every 6th person on the planet now. Men are more likely to suffer from lung cancer, and women from breast cancer. At the turn of the 4th century BC, Greek scientist Hippocrates compared an advanced breast tumor with a crustacean because it looked like the swollen arthropod and its vessels resembled claws. Humanity is searching for the fastest possible way to cure cancer. Russian scientists have come close to a unique way of detecting and treating cancer at its early stages.
People, doctors, biologists, drug developers have always been thinking about how to deliver medicines specifically to the required spot. How to fix this breakdown that occurs in the body. How a drug can treat a disease without affecting anything else in its path. The medicine should not affect other organs and systems. Now, we are talking about creating effective drugs for oncological diseases and degenerative disorders. And we, as chemists and biologists, have to specifically deliver a drug to this target.
Luba, tell me why the drug should be delivered to a specific place in the body.
Of course, I'll tell you. Once in the body, the medicine dissolves in the blood system and spreads through the blood vessels. The medicine heals one organ, but it can damage the others. For example, an antibiotic. It kills harmful bacteria and treats people for infections. At the same time, it destroys beneficial bacteria required for normal bowel function. If the drug gets into a specific part of the body and does not affect other organs, it minimizes the side effects of such a drug. The pill only benefits the patient. This is called targeted drug delivery. I'm sorry, I might have stressed the wrong vowel.
Our department has always worked with macroheterocyclic molecules, most importantly, photoactive ones. These are photosensitizers. Photo means light, to sensitize, to absorb light, and these molecules are special. These molecules include hemoglobin, chlorophyll, or green plant pigments. Firstly, these are natural compounds. And secondly, it turns out that they themselves can target and detect tumor processes. I'll explain. When these molecules enter the bloodstream, they quickly pass healthy organs and tissues, but they are accumulated in the tumor nodes. We may call it selectivity. That is, these molecules remain in the tumor nodes longer than anywhere else in the body, and doctors may use this retention time for diagnostic purposes. When the doctor sees the glow of the spot where photosensitizers have accumulated on the scan, it means that the doctor sees the localization of the tumor, its margins, its size. The doctor can get this information straight away by injecting the drug. We can see where the tumor is, what type of tumor it is, its size. Moreover, this way you can diagnose even a very small, tiny tumor.
Hold on. I will explain what chlorophyll is. Chlorophyll is a green pigment that turns leaves of plants green. Everyone has ingested chlorophyll with food, as it is found in leaves of any green salad. This substance is a source of beneficial antioxidants. At the end of the 20th century, scientists discovered that chlorophyll was rapidly excreted from all tissues, but it lingered in tumor cells. Since then, oncologists and chemists have been trying to develop the best way to diagnose cancer using chlorophyll. This technique was called photodynamic therapy, PDT.
Nuclear medicine is in the spotlight of the world community now because radionuclides, due to the high energy they emit during alpha, beta, and gamma decay, can both diagnose a tumor and treat it. The biggest drawback of chemotherapy is its high toxicity. If we deliver the drug directly to the tumor, the toxicity will be reduced by targeted drug delivery. And if chemists can attach so-called chelates to our molecule, chela, meaning claw, the molecule will wrap its claws around radionuclides, and then our porphyrin chloride will carry this radionuclide straight into the tumor.
Technetium is a radioactive transition metal. It is used in medicine to localize tumors, as the technetium isotope accumulates in their tissues. Lutetium is a dense rare earth metal. Once inside the tumor tissue, it can destroy the cells. Now scientists will tell you about how these elements can be used in cancer treatment.
There are some radionuclides that, due to their powerful energy, can destroy cancer cells. Then we can carry them with the drug as well. For example, we're currently working with technetium. Technetium radionuclide is used for diagnostics. And there are, for example, such chelates that can carry lutetium. And lutetium has a high radiation exposure, but lutetium can destroy a tumor cell. And this property can be used in therapy. In other words, on our platform, we can create diagnostic drugs, chemo drugs, radioactive pharmaceuticals, RAP. We can carry out diagnostics. So this is a universal platform, as we say, a multi-purpose one. We're creating a platform for making different drugs, but all of them are targeted. They won't disperse throughout the body. They won't circulate in the bloodstream for a long time, irradiating and poisoning the body. After 2 years of work, we've made a molecule for creating radiopharmaceuticals, which was tested at the Siberian State Medical University on cells with technetium radionuclide. And that test was very important because the radionuclide was already embedded in the molecule and it showed accumulation in cells. In Moscow, this molecule was tested on animals, but without the accompanying radionuclide, because the work with radioisotopes had some limitations.
We tested the biodistribution of the compound on animals with lung tumors and lung carcinomas. This compound showed high accumulation in these tumors. In my experience, that was the first time I saw such accumulation in a tumor. There was very high fluorescent activity. The accumulation in the tumor was 3 to 10 times greater than that of currently existing dyes.
We need to collect statistics. When 1 mouse out of 2 dies, it is unclear what caused the death. But if 80 out of 100 mice were treated and the outcome was good, this result is reliable. Therefore, we need statistics. We need large groups of animals and a lot of drugs to be tested. So now, I would say that the scale of study is important. That is, The scientific research phase is over. We're now at the stage of scaling and verification. Chemists created this drug, and now it's up to biologists, who should find out what happens to animals, how the drug accumulates, and what happens to the tumor of this animal. The result can be quantitatively measured with a tumor size and speed of its reduction, or TGI, tumor growth inhibition. If the tumor growth is inhibited intensively, then we have achieved our goal.
Luba, show us a pill with a targeted drug delivery property.
This is how I see the targeted drug delivery.